So thank you very much for the introduction and for the opportunity to present my work here. And with my presentation, I'm hoping to bring a bit more diversity to the 3 R's training webinar series. And by diversity, I mean that when we talk about animals used in research, most of us would think of a rat or a mouse kept under laboratory conditions. But research is being done on many different species, also of wild animals in a field. So for example, in ecology and conservation biology. And this research is important because many species are being threatened with extinction due to adverse effects of human activities. And the information that we gain from studying wildlife populations can inform decisions that will ensure species survival in the future. And in research on wild animals, we might be using different methods. So you can see here capture, blood sampling, marking, or fitting animals with various instruments. And when these methods are well implemented, and this research might have minimal impact on the animals. But we need to be cognizant that there have been reported some animal welfare issues with these methods. So I would like to provide here a few examples. Let's start with capture. So because wild animals are not accustomed to being handled by humans, as some lab animals might be, just capturing itself might be extremely stressful for them. And for example, Catet et co-authors showed that grizzly bears that had been repeatedly captured had significantly differed in their body condition compared to bears that have been captured only once. And another study by Roth and others reported changes in behavior of polar bears several days after capture. And in some cases, the stress of capture can lead to so-called capture myopathy, which is metabolic muscle damage resulting from extreme exertion, struggle, and stress, and it's often lethal. And some species are prone to capture myopathy more than others, so we need to be especially careful when we are capturing deer, rabbits, or even birds. And apart from stress and impact on behavior, capture can also result in a physical harm. So these damages can range from skin abrasions to broken limbs. And trapped animals are also vulnerable to predation and environmental conditions such as excessive heat or cold. Now let's move on to blood sampling, which is one of the most common methods for obtaining either DNA or samples for physiological or toxicological analysis. And blood sampling could be difficult in small animals, and even though it's considered relatively safe, there have been studies showing that it might affect survival of some animals. Another common practice in wildlife research is toe clipping, used for marking animals or for tissue sampling. And it is mostly implemented in amphibians, as you can see in this photo, but also used for marking reptiles or small rodents. And this is a potentially problematic method because it has been reported that toe clipping can reduce survival rates and affect the movement ability of animals such as lizards and frogs. Another common method of marking animals is with tags or bands, and these are made of different materials, most commonly metal or plastic, and they usually have on them some alphanumeric code so that you can distinguish individual animals or groups. And in the picture, you can see a king penguin with a tag on its flipper. And there's now detailed evidence showing that such flipper banding can severely damage the tissues. And also there is a drag effect resulting in a higher energy expenditure when birds are moving through the water. And this can then result in lower efficiency in foraging, since the marked animals require longer foraging trips, as well as in lower survival and then lower breeding success. And another research method is the attachment of instruments, for example, radio transmitters to monitor the animals. And similar to tags, radio transmitters might affect behavior and energy expenditure. And other important factors are the method of attachment and weight of the instrument. So if the radio transmitter is attached by glue, this might cause skin lesions and abrasions. And sometimes the radio transmitters are not attached, but implanted into the abdominal cavity of the animal. And there have been, in the meantime, published several studies, for instance, in snakes and fish, showing that these implants can cause inflammation. And other studies even reported that some of these implants have malfunctioned and it caused death. And this has been reported in badgers, bears, and lynxes. And lastly, also lethal methods are sometimes used in research on wild animals. And this is also common in cases where this would not be necessary, such as when we want to collect data on the species diversity or on abundance. So these have been some commonly used methods in wildlife research that might have some impact on animal welfare. But why is animal welfare actually important in wildlife research? So anyone conducting research on animals, regardless if it's in the lab or in the field, has the ethical, scientific, and legal obligations and incentives to minimize harm to the animals. So let's first have a look at the ethical considerations. My background is in ecology and species conservation, and the focus in my field has traditionally been on preserving species, populations, and ecosystems. So this is a quote from Michael Soule, who is considered the founder of conservation biology. And he writes that conservation is engaged in the protection of the integrity and continuity of natural processes, not the welfare of individuals. However, as we witness a growing demand in the society for humane treatment of animals used in food production or in biomedical research, the sensitivity to research and management activities affecting wild animals has grown as well. And you can see that the ethical treatment is really vital for maintaining public trust. So if the public perceives that animals are mistreated or the researchers are using excessively invasive or lethal methods, it can lead to backlash against scientific research. So for example, there was a widespread public outcry surrounding the southern elephant seal hot branding for a research project after a video footage was released showing poorly marked individuals and animals with unhealed wounds, and the study was stopped by ministerial intervention. And there are also voices within the scientific community expressing the need to consider ethics in wildlife research. So for instance, the editors of the journal Biological Conservation reported in 2016 that they had decided to reject several manuscripts for publication on ethical grounds because they considered that the authors used lethal methods that were unnecessary and appropriate. So ethics plays an increasingly large role in any field of animal research, including wildlife research. Now let's look into why we should care about animal welfare for scientific reasons. So going back to my examples of animal welfare consequences when you capturing, for example, bears, if we are interested in behavior, so it the capture itself might affect how the animal behaves. So the results that we get from such a study might be misleading. And also the study that I mentioned with tagging penguins, it has been shown that actually studies, including those that study the effects of global warming on and their effect and its effect on seabirds, may be inaccurate because it's impossible to distinguish the impacts caused by the marking from the global warming impacts. So in summary, poor animal welfare is not only bad for the animals, but also for the science. So if we want to make sure that our data are accurately reflecting what is happening in the wild, we need to minimize animal welfare impact. And lastly, there are also legal obligations to minimize harm to animals used in research. So I'm sure that all of you are familiar with the 3Rs principles that have been proposed over 60 years ago, and now they are implemented in many legislations worldwide. The 3Rs have been developed with lab animals in mind, which is reflected in the implementation strategies. So for example, we can replace animals with cell cultures. We can reduce the number of animals used through breeding optimization, and refine the experiment through better housing conditions or handling. So these strategies work well with lab animals potentially. So cell cultures could be successfully used instead of a mouse to test a toxicity of a new compound. But such a replacement strategy would not work in wildlife research. So we cannot use cell cultures to find out how many different animal species visit a watering hole or how animals are moving within a landscape. So because wildlife research has specific conditions, it may not be as straightforward to implement the 3Rs principles as perhaps in the lab, but there are some strategies that work. And I outlined some of them in a literature review that I published in Wildlife Biology in 2020. So as a replacement, we might use archived museum samples. As refinement, we can implement anesthesia or improve trapping methods. And as a reduction, we can collaborate with other teams. So for example, if there are taking blood samples to assess the genetics of the animal population, we might use these blood samples also for health assessment. And so that we avoid sampling the same population and potentially the same animals twice. And one of the most straightforward ways to implement the 3Rs in wildlife research is actually to use non-invasive research methods, which means methods that do not penetrate the skin barrier. So I will now go over some examples. If we want to conduct a genetic study in frogs, so instead of toe clipping that I mentioned before, to collect the tissues, we can use either skin or buccal swabs. Instead of capturing animals and taking blood or tissue samples for genetics or health assessment, we can collect something that all animals leave behind, namely their feces. And feces are actually a great material also for dietary analysis, where they can replace lethal stomach content sampling, because we can identify the animal's diet composition through metabarcoding. Also in physiology assessment, instead of blood samples, we might use hair, feces, or saliva samples to assess hormone levels. And if our study design requires that we are able to distinguish individual animals, instead of invasive marking or attachment of instruments, in some species, it is possible to use natural markings, such as scars or some other patterns. So such Such photo identification can be done manually, but nowadays there are actually available several software packages that implement deep learning algorithms and are able to distinguish patterns that might not be as easily visible to the human eye. And lastly, if we want to detect species to estimate the species richness in an area, instead of capture or direct observation in the field, we might use either camera traps or the so-called environmental DNA. And the principle behind the environmental DNA is that every organism, as it moves through the environment, it sheds some cells, some DNA. So, for example, in form of mucus, or when animals eliminate waste, or when they are drinking from the water. So I could actually go to take samples from this watering hole, and I would be able to detect that zebras and giraffes were drinking from this water just based on this water sample. Environmental DNA has been mostly used in aquatic environment, for example, detect fish or the species that are drinking from small water reservoirs. But it has been recently also taken to the land. So it's possible, for instance, to detect beetles just by collecting their DNA traces from the vegetation in the area. So these were a few examples of non-invasive methods, and you can really see the wonderful creativity that has been put into developing these methods, which is amazing. And you might be thinking that everyone is probably using these non-invasive methods whenever possible. But this is actually not the case. So in 2019, I conducted a review of published wildlife genetic studies, and I assessed whether they used a lethal, invasive or non-invasive DNA sampling method. And I found 181 relevant studies. And out of these, that I found that in studies on amphibians, only 14% used non-invasive research methods. In birds, 8%. Carnivores were exception, there it was 81%. But mollusk none, and in rodents only 12% used an non-invasive method. So on average, only across all these different types of animals, only 22% of studies use a non-invasive technique. And important to point out is that for all these types of animals, there's at least one, probably more, validated non-invasive techniques that could have been used instead. What's happening? Why is the implementation rate so low? I conducted a pilot study that I published in 2021, reporting on a survey among ecologists that work with animals. And I had 107 respondents from 23 countries covering different career stages. And out of 170 respondents, only 39 knew the 3Rs principles, and only 22% used a non-invasive research method in their latest study. And when I asked, what do you consider as the barrier to implementing non-invasive methods in ecological studies, the most often listed reasons were financial constraints, but the second common barrier mentioned was the lack of awareness of existing non-invasive research methods. And this is one of the reasons why I have actually started developing some resources to help other wildlife researchers to implement more non-invasive methods in their work. So I have created an informational website that is available at the 3rswildlife.info. And the website has several sections. So first, there's some background information about the importance of wildlife research, potential animal welfare issues, the 3Rs principles in general, and also how to implement them in wildlife research. And the main section actually consists of example studies that either developed or implemented a non-invasive method. And they are now available 8 different categories: dietary analysis, ecotoxicology, genetics, health monitoring, individual identification, morphology, physiology, and species detection. So when we have a look at, for example, physiology, there are subcategories for invertebrate and vertebrate animals. And under each subcategory, for example, under birds, you have listed the different studies that either developed or implemented a non-invasive method, and which can then be used as a guidance or as an inspiration for other researchers when they are designing their study. And a recent addition to the website that has been made possible by the generous support of the Animal Protection Commissioner of Berlin are short videos demonstrating some of the non-invasive research methods. So far, there are 4 videos available. So one is on fecal collection for DNA or dietary analysis. Second one is on mucus swabbing in slugs and snails for DNA collection. The hair sampling without capturing the animals. And the last one, skin swabbing for DNA collection in frogs. And this video we can watch now together.
Hello, and welcome to this instructional video on skin swabbing in frogs for DNA collection. In this video, we'll show you how to perform this technique. After capture and prior to swabbing, animals should be placed in individual containers to keep them safe. To begin, put on your powder-free gloves and moisten them with a pond or clean water to avoid damaging the frog's skin. Hold the frog firmly but gently. Do not press the frog's abdomen too much, as this might cause damage to the internal organs. Next, gently rub the sterile swab across the frog's skin, being careful not to apply too much pressure. The best areas to swab for DNA are the abdomen and thighs. Repeat the swabbing 5 to 10 times in each area. Once you've swabbed the frog, carefully place the animal back into the container. Afterwards, put the swab into a labelled sampling tube and store at -20 degrees Celsius. Remember to change your gloves before handling another frog in order to avoid cross-contamination between samples and accidental spread of pathogens between the animals. And that's it. You can now release the frog at the location of capture and send the swabs to a lab for a DNA analysis. Thank you for watching this video. We hope you found it informative and helpful. Good luck with your research.
This was just one of the videos, and if you want to see the other ones, you can find them either on the informational website or on YouTube, where you can actually watch them in a 4K quality, and also choose captions in English, German, French, and Czech. And this brings me already to the end of my presentation. So to summarize, wildlife research is essential for effective species conservation and management. But unfortunately, some of the research practices can be detrimental to animal welfare. So it is imperative to minimize the welfare impact on animals used in research, and as I mentioned, for ethical, scientific, and legal reasons. And that is why it's important to raise awareness of these issues and to create resources to help implement more non-invasive methods in wildlife research. So my take-home message is, when we talk about the 3 R's principles and their implementation, let's not forget the animals outside of a lab. And before the very last slide, I need to thank several organizations for their kind and generous support of my work, and in particular the Animal Protection Commissioner of Berlin for making the creation of the instructional videos possible. So thank you for this opportunity to present my work, and thank you all very much for your attention.